Click for IB Biology. We're going to be looking, amongst other things, at the directionality of synthesis, of transcription and translation. We'll look at promoters and transcription factors, and also in post-transcriptional modification. There's a blank PDF of this presentation down in the description. You can annotate with that with your own notes. Test yourself with the 10 questions, and the answers are there. And then if that's not enough, you can then post my AI prompt into a chatbot of your choice, and that will generate an individualized study program for you. Okay, so the first thing is the direction in which transcription occurs is noted as 5 to 3. Now this phrase 5 to 3 is referring to the building of the messenger RNA molecule. And that relates to the idea that the RNA polymerase, which is here, is adding the 5' carbon to the 3' carbon. So if we take at the first two nucleotides here, then the 5' is on the second nucleotide, that's this one here, is added to the 3' of the first. So that one here is added to that one there, and then the polymerase works its way down the chain in that direction. So the next one, of course, would be that the 5' of the third nucleotide, this one here, would be added to the 3' of the second, and so it builds the chain in a 5 to 3 direction. It's referring to the action of RNA polymerase. Now this does leave us with a 5' messenger RNA at this time, at this position, and a 3' messenger RNA at that end. So the nucleotides at the ends are in that direction as well. Now translation refers to the movement of the ribosome along the messenger RNA chain, and the ribosome will move from the 5' messenger RNA end to the 3, reading the codons as it goes in sets of 3, what the 3 bases, which are the codons. So that's also 5 to 3. If we take a look at the beginning of a gene, you'll remember that we noted that the start of a gene is located upstream is the promoter. And these are often referred to as Tata boxes, in which on one of the polynucleotide chains of the DNA, it will have the sequence TA, TA, A. That's a common one. And then the complementary sequence, of course, would be A, T, A, T, T. Now these sequences are multiples, and they can repeat themselves. And these promoters mark the beginnings of genes, where the helicase and the RNA polymerase begin their action. These promoters are essential for allowing the access and the action of RNA polymerase. So the gene will not be transcribed unless the RNA polymerase can get to the gene, and it does so by going through the promoter region. Now the promoter region can be affected by the cell environment, factors either in the cell or factors from outside the cell, the environment in its broadest sense, by what are called transcriptional factors, and these are proteins. Now these proteins can be activators of RNA polymerase, or they can be suppressors, and they do that by interacting with the promoter region. So this is a way in which genes can be regulated, being switched on or not, on and off by factors present within the cell. Now there are a number of other items that we need to know in our higher level study of protein synthesis. And one of them begins here. This is an image of a chromosome, and the blue region here represents a gene. Now, there will be multiple genes along a chromosome, of course, but then there will be regions in between these genes, and these are called non-coding. Now, the gene regions produce messenger RNA. So genes, they produce messenger RNA. But the non-coding regions can be the promoter Tata boxes, or they can be the regions that locate the genes for tRNA and rRNA. So although they're called non-coding, they're actually producing either they're a control promoter or they're producing tRNA or rRNA. So they don't do nothing. So let's return to our chromosome. Now, the chromosomes are protected from the cell environment and from damage and from cell division by the presence on either end of the chromosome of what are called telomere structures. And the telomere structure are long sequences of bases. I've illustrated one here. So this is a telomere sequence. And again, you'll notice that we have this predictable sequence of CG, 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 TA, AT, AT. And this can actually, the reason I've bracketed this is because this can be very, very long, especially early in the life of a cell. And then there's a long sequence of Gs on the end like this. And this loops over like so and protects the chromosome from the action of enzymes called nucleases, which will actually break down DNA and other nucleotides within the cell which need to be recycled. So it's a way of protecting the cells. If we move on to then the messenger RNA, when the messenger RNA is first produced in transcription, the long sequence of the messenger RNA, the copy of the gene, which I've drawn larger here, it also is protected from being broken down by what are called caps and tails. So there's a cap at one end and a tail at the base. Now the cap region, the 5' end, is made up of phosphate, phosphate, phosphate to guanine, and that protects the end of the messenger RNA at the 5' end. Whereas at the 3' end, there's a long sequence of adenine, 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 possibly more, protecting that end from damage from enzymes. So we can protect both our DNA and our messenger RNA with the addition of these telomeres in DNA, caps and tails in messenger RNA. Now, the first structure produced by transcription is referred to as pre-messenger RNA. And within the sequence of codons, there are bases within these which are known as introns, and here they're in white. What happens is that the introns are removed, and then we splice together the exons to give us what is called the mature messenger RNA. And of course, then that's translated to form a polypeptide. Now, the interesting part now comes is that with the exons, they can be sliced together as I've shown in my diagram, or one or more exon can be removed, or sometimes the order of the exons can be changed. Now, the consequence of that is that we will change the protein. And so our proteome, which is the sum of all the different proteins within a cell, is far greater than the number of genes that we have. Humans have been noted to have about between 20 to 22,000 genes, but we have many, many more proteins known as our proteome. This is our genome, but here we have our proteome. And this leads to a greater diversity of proteins that can operate within the cell than would be predicted from the number of genes that we have. We're now going to build a model of translation at higher level in the next video, subscription to the site, and the playlist, of course, for you to look at.